So.
Who can see introducing the non-ardent self?
Yes, we can.
You can see it? Everybody see it?
They're all muted. So
Raise your hand if you can see the slide. Yay. All right. So many of you probably know me. I know many of you, but maybe not all. And so we had, Jen and I had a little chat, and we decided I would retire. I would introduce myself. And so I'm not a native Ardenite or a native Delawarean. My wife and I moved here in middle age. Mary Brent Whipple, MB. We moved to 210 Hillside Road in 1981 from Burlington, Vermont. And then I retired as a DuPont pensioner in 2001. So I'm a retiree. At that same time, and not unrelated, we moved to the Ubersax house at 2145 Veal Road in Ardencroft.
Apple computer, he had Microsoft.
Microsoft.
In order to keep active in retirement, I began commuting to Center City, Philadelphia, to Thomas Jefferson University to do biomedical research. And here's my official titles. If you need to unpack the academic jargon, feel free to ask, but it doesn't really matter. That's just the role I have at TJU. So today we're going to talk about the little brain in the heart. Who knew there were neurons at the heart? But there are. And you're going to find out why they're interesting. So we published a paper on the topic, and it went incredibly viral. I was shocked. I've never had anything like it happen. You know, we labor away in the vineyards of knowledge and fight with editors and get papers published, and sometimes the number of people that cite it or view it or look at it is just about zero. It's pretty damn terrible. But sometimes, and this time, it got a huge amount of attention. It just, people, all kinds of friends and neighbors were sending me links saying, I just saw an article about your paper. That was really crazy fun. And so many different outlets wanted to interview us and put the interview together into a piece. And so it was a really busy time in the spring and through June. So I want to tell you about it. I'm going to tell you about this stuff that was in the paper that got a lot of public attention. So since it got public attention, maybe it'll be of interest to you. So I'll share this with you and we'll find out. So I have an outline of what I think I need to tell you to make this work. You need some background on how the brain works in order to figure any of this stuff out. So I'm going to start out with some background stuff. It might put you to sleep, or you might go, that's really interesting. Now I know something about how the brain is organized. Isn't that cool? And then I'm going to tell you about an NIH program. So the NIH is National Institutes, plural, of Health. So there are all these different agencies, as they call them. There's the Neurology Institute, there's the Mental Health Institute, there's the Drug Addiction, and so on and so forth. And then there's the Director's Office. Francis Collins keeps some of that money back for himself and creates a common fund to do stuff he thinks is of more general interest or importance. And so he created, with his common fund, something called SPARK, and I'm going to tell you what SPARK is. And I'm going to tell you what the teamwork it took in order to accomplish this, how it was a interdisciplinary effort and an effort across collaborators across all kinds of places. Auckland, New Zealand, San Francisco, UCLA, Burlington, University of Central Florida, and so on. And then we'll go through the paper itself. We'll just walk through and look at the figures and say, so why was this interesting? And if we have time, I may very briefly speak to what this enables, how it gets to be translational, how it gets to be used. We'll see if we get that far. So here, let's start by getting a few facts into play for background. The brain is in your head. It just sits there, but it's not in splendid isolation. So how does it interact with the world? How do you know what's going on in the world? And how do you get to do anything? It's through nerves. And in particular, the cranial nerves are really important for how the brain interacts with the world. And the one we're going to really focus on is called the vagus nerve, cranial nerve 10, which innervates the heart. And it's that activity that actually keeps the heart healthy. And that's really going to be the focus. So this is a Leonardo da Vinci thing that is a thing of beauty. It's amazing what he did. That guy was just a genius. And so what you have down here in the lower right quadrant is the brain is in your head, right? There's the skull cap and cranium. There's the brain, eyeballs, optic chiasm, all these cranial nerves by which the brain interacts with everything else in the world, so that you know what's going on and you're able to do something about it. And that all sits down into the head through all these holes in the bone that are called fossa. There's these horrible things called fossa that you have to try to microdissect and figure out where all these nerves run in order to get where they need to go. So there's that business. And then up here, this guy looks pretty dead, wouldn't you say? How did Leonardo keep his cadavers going for very long? This guy looks pretty bad, but Leonardo got it really right. Really, really right. And for one thing, the brain itself, which is this business up here, he's got these holes in the brain, right? Which we now call ventricles. And he's got them more or less right. He's got lateral ventricles, he's got a third ventricle, he's got an aqueductus sylvius, he's got a fourth ventricle. Pretty cool. You know, considering the dissections he was able to make, he actually represented a lot of things that are true and important. But all these little gizmos here that are coming out, the eyeballs, and to muscles of the face, and to tongue, and to muscles of mastication, and all this stuff, oculomotor stuff, you know, these are more or less right. I got it pretty damn right. Pretty shockingly right. I guess we already knew he was a talent. He is quite a talent. And for some reason that I cannot figure out, maybe one of you have an insight, in the same drawing where he's got all this stuff going on with the brain, he represents the male reproductive system. Here's the testicles, here, the penis, vas deferens. Why does he do that? Put it in the same frame. Who knows? Maybe one of you have a special insight. But, so in a more modern representation, here's a human brain, and the different cranial nerves are tagged for the stuff they do. And then here's a little more abstract representation of that. Oh yeah, I need to tell you. So the brain is not just in your head. Yeah, he didn't get it. This is something he didn't get. He's got the whole brain, it's just this big lump. But these days we like to divide the brain into forebrain and brainstem.
You need to know all these, Jesus.
Into at least forebrain and brainstem. So this would be, you know, this is frontal cortex here, and the different lobes of the cortex are all this big massive stuff. And then these guys lumping out to the side are cerebellum, which is not cerebral cortex, but something else entirely. And then this business here, all of this through here is called the brainstem. And these cranial nerves connect in and out in specific spots into the forebrain and into the brainstem. Sure. And here's a more abstract representation of that. So we'll talk about what each of these do in a maybe we will, maybe we won't. Olfactory is smell, right? Optic is vision. Here comes the optic nerve and the optic chiasm and optic tract. And ocular motor moves your eyeballs around. So does trochlear. So does abducens. Move your eyeballs in different planes. Fifth cranial nerve, facial. Muscles of facial expression and so on. This moves your tongue. This is your guy wags your tongue. Here's audition and balance. The one that we really care about, though, is this one here. This is the one we're going to be talking about, vagus 10. I love this. I don't know if this makes you smile like it makes me smile, but all medical students learn the cranial nerves with one or the other of these, what do they call them? An old Olympus towering top, a Finn and German viewed some hops. What do they call that again?
Mnemonic.
Mnemonic. Most of the ones the medical students like are not printable here. They're pretty nasty. But you get a mnemonic that you can use to track it, you know? So that's the order. T, T, you know? And that's how you learn them. I don't know anybody who didn't learn them that way. I certainly learned them that way. And I still do it, you know? I go, oh, which one is trochlear? Two. It's terrible. But all these things, like, you know, here's olfactory, here's vision, and so on. So I think it's hilarious. Maybe somebody could draw a prettier one, but I think she's cute. And oops, then this one actually shows the actual, you know, innervation, the fields of innervation, sensory and motor, and what the functions are of each of these nerves. And the one we're going to be focusing on is the vagus, which comes out of the medulla, out of the brainstem, has sensory and motor components, and innervates the viscera. It's the one, as you saw in Leonardo da Vinci's drawing, it's the big pipes that hang down out of the brain, go into the body. That's what this guy does. And you can see it's getting around here, right? Lungs, heart, esophagus, stomach, intestines. And so I should mention that there's something called, this is the peripheral nervous system. There are two kinds of peripheral nervous system, somatic and autonomic. So somatic is just directly interacting with like muscles and skin and pain and joints and stuff like that. Autonomic is the part that regulates the organs, the viscera. And so here we see the parasympathetic side having a vagal outflow here that goes heart and decreases heart rate contractility and heart rate and contractility of the heart, and lots of other things. The vagus is a busy guy. I guess it is root in ancient languages is wanderer, and it gets around. And so we're going to talk about the vagus more in a minute. So here's the vagus nerve anatomy. And so you can see here that here's the peripheral part, which is what we've been looking at. Here comes the nerve. It's got all these branches to the pharynx and larynx, lung. And here are all these cardiac branches. That's the part we're really interested in. And its origin, so a little secret is that awful lot of central nervous system neuroanatomy is based on these nerves. We find out where these nerves, where the sensory aspects of these nerves land, and where they originate, where do they come from, what neurons give rise to them. And that's a start. That gives you an anchor to the periphery. You know what the periphery is doing. Oh, that's the heart. So where are the neurons for that? And heart sensation or blood pressure sensation, where is that landing in the brain? That gives you an anchor. And then you work from there. You try to figure out, okay, so now I know who's talking to the heart, but who are these guys talking to inside the brain? And you try to do a connectome and march your way through the brain in terms of the sequence of connections. So an awful lot of neuroanatomy is basically rooted in starting from the peripheral inputs and outputs. And so what you see here is that so this is the brainstem. We were looking at the brainstem a minute ago, right? And we're looking down on the ventral surface, the bottom of the brainstem. And so this is called caudal towards the tail, and this is called rostral towards the nose. And in that plane, everything is like a column. There are these long columns. Here's the seventh nerve, facial, fifth nerve, trigeminal, and so on. But vagus stuff is coming out of long columns like this. And I'll show you something I did and I'm very proud of. Back in the day, we built a digital brain atlas and we were able to map the location of every neuron that contributes to the vagus nerve outflow, and 2 rats. And this is every neuron. And there are 2 nuclei that contribute to the vagus nerve. One's called the dorsomotor nucleus, huge long column of neurons. And this is the nucleus ambiguus, G-U-U-S, A-M-B-I-G-U-U-S, but it's plenty ambiguous. And here's a second rat. You can see it's got a very, very similar morphology. And here they are put together in one framework. So we did that in DuPont in 1988, way ahead of our time. And then Siemens bought all the technology and the postdoc who did the work, and we stopped. Damn it. But it was cool while it lasted. So that is what the population of neurons are that give rise to the vagus nerve. And then they project to the heart. And so the heart, strangely, is a little bit upside down. So here, the top of the heart that you would think would be the apex at the top is actually called the base of the heart. Because developmentally, when things grow up embryologically, the heart begins with cells that migrate down in the great vessels and then starts building itself out from here, this direction. And so this gets called the base because that's where it started. And then here, down this little pointy guy, which you think, well, surely that would be the base because that's at the bottom. No, that's the apex. So just so you get oriented. And so as it happens, interestingly, the vagus nerve, being part of the parasympathetic nervous system, does not actually innervate the heart directly. It innervates neurons that are located at the heart. There's a whole population of poorly understood neurons through which any vagal influence has to express itself. It cannot directly influence the heart. It's got to influence this population of neurons that in turn do whatever they do to the heart. So mapping the projections for the dorsomotor nucleus we were just looking at as they go to the heart and land on this population is key to getting any kind of hint of how the vagus actually influence the heart. Right? Is that making sense so far? Nod your head up and down. So I couldn't resist just sticking this in real quick. This is stupid stuff. But just so you know, neurons are, I'll show you in a minute how it's thought that they're individual processing units. It's not like syncytium. It's not like they're all linked together in one big connected web. They are connected, but they're connected through chemical interactions that release transmitters. And they release transmitters based on electrical activity that creates these action potentials, they're called, that cause chemicals to get released and to get a so-called synaptic cleft between the presynaptic and postsynaptic neuron, and that influence the postsynaptic neuron by the release of the chemical. And then you get all these neurons linked together and they go into networks and all of that. So there's that whole business for understanding how brains work. This is what neurons look like. Is that cool? I'll tell you a story. This is called Golgi staining. So in the 19th century, 1880s or so, a guy named Golgi in Italy found out that if you take a tissue section and you wash it with a silver solution and then rinse it out, that it stains like 5% of the cells present in the tissue section, but it stains them thoroughly, really thoroughly. So you get a picture of everything. You get a picture of the cell bodies and the dendrites and little axons coming off. And it's a quite amazing picture. Doesn't that make your skin crawl to think you got all those little spiders in your brain? but that's what neurons actually look like. And so he did good work, Golgi, but he didn't do quite as good a work as a Spanish guy called Ramon y Cajal. And so Ramon y Cajal was a master artist, and he took the Golgi method and he drew incredible drawings. In this case, I believe of Purkinje cells in the cerebellum and the axon sneaking out here. And so, wow. Yeah. And Cajal invented the so-called neuron doctrine. Neurons stand alone. They're only influenced through not being part of another neuron, but through external influences from neurons and other cell types. That was the neuron doctrine. And Golgi hated it, and they hated each other. But of course, they jointly got the Nobel Prize and gave highly hostile talks about one another. So that's it. People didn't always get along, even in the 19th century. And so let's talk about Spark. So we're part of this large NIH initiative. It's aiming to learn how the vagus keeps the heart healthy. So this is, and even how to reverse heart failure. So this is a thing that's been known for quite a long time, is that there's a correlation between the activity of the vagus nerve with respect to its innervation of the heart and how healthy the heart is. If you have a so-called low vagal tone, if the vagus is not active enough, you will get heart disease. And there's been a considerable amount of success with actually reversing heart disease by elevating vagal tone. There have been clinical trials like crazy, but they didn't do a good job. They're poorly designed and they have very uneven results. So they have results, but they're not consistent enough to go clinical. And and nobody knows how to identify either the stimulation parameters of the patients that could benefit. Some can, but nobody knows which ones. And so Spark wants to find out. The Spark stands for Stimulating Peripheral Activity to Repair Conditions in Peripheral Organs that the vagus innervates. The vagus can be a health-giving good guy. How do we find out how that works and emulate it? So for example, if you were to understand the neurons at the heart and how they're organized and what they do, could you implant a little microchip, a little microcircuit on the right neurons and stimulate them in the right way and make a healthy heart, for example? So let's look at some. I hope these links will work, I pray. Yes. So this is Spark. Can you see it? The common fund is stimulating peripheral activity. And there's our heart. That's the one we did. And so this link, and I can share all.
Jim, we can just only see the slide that says Spark. It doesn't show it. We can't see the heart.
Oh, you don't see the heart up here?
No, we don't.
Okay. Do you see this?
We see all the hyperlinks.
But you don't see the hyperlinks product?
No, we don't.
It's on my screen. How come it's not on your screen?
You may have to share your whole desktop. Otherwise, it's just sharing your presentation.
Yeah, you may have just shared your presentation and not your screen itself.
Okay, I'm going to go share the whole desktop. That must be the problem. I bet you're right. Makes sense. See it now?
Yep, that's it.
Yep.
You see it.
All right. So that's the Spark website. You learn more about Spark than you want to know. And here's the next one. Welcome to the Spark portal, where you can dig around and get more data than you ever thought you wanted. So this is all kinds of Spark information in here. Explore data sets from your, you are in luck. Here's one of the science focus articles on our little work on the brain, on the heart brain. And here's a cool map that you can interact with. It shows you all the ways the vagus nerve interacts with rats, with the periphery of the rat. And so if you're curious about this, this is interactive and you can spend a joyous time, exploring it. Since all this is recorded, you can get to these links. Maybe you'd like to, maybe you wouldn't. All right, so so we set out to discover and map all the neurons at the heart and make a comprehensive picture, three-dimensional picture of their distribution and their molecular identities in order to get some idea of which ones you might want to approach to stimulate to make the heart healthy. So let me show you the paper that got all the attention. This is it. Whoops, what happened? I had it. It thinks it needs to reload. Okay. Okay. So this was a big collaboration with a lot of different groups, and we started out with a heart, and we embedded the heart in something steady while it was being cut. And then a company in San Francisco that was one of the collaborators, 3Scan in South San Francisco, had a fancy method. It was really crazy. He called it knife edge scanning microscopy, where they had diamond knives that are transparent. You can see through the diamond knife. And so as the diamond knife would be taking a thin section off the heart, you had a high resolution camera imaging the heart at a resolution of a 0.5 micron by 0.5 micron. And so all of that stuff all that data was being streamed. And then from there, we would go to building a stack, a so-called stack of those images. And then we would map all those position of all the neurons that were present in each tissue section, and then we stacked it back along with the contours of the structure of the heart and built a 3D model of the heart. So that's what we did. And it was a big collaboration with people from TJU and people from all over all these different places that contributed, including people in New Zealand and San Francisco and so on. So here's the thing I just showed you, how we built the models, and this is what we got. And so the yellow business are the neurons. They're all the other stuff is different structures of the heart: left ventricle, right atrium, right atrial appendage, and so on. And so, but the distribution of neurons is shown here from different points of view, and thinning out the other structures so you can see it better. And we're able to take the whole heart and cut it into slabs to see where the neurons are within a particular region of the heart. This is looking down onto the heart from on top. And so one of the big surprises, which I'll tell you about in just a second, was where these neurons are. And that was one big surprise. So it used to be thought that all these neurons were, such as they were, had two properties. One is they were all located on the base of the heart around the major vessels. So the pulmonary veins and arteries and the aorta and all that stuff are all going in and out from one specific location on the base of the heart. And all the neurons were thought to be just positioned right around those vessels, because that's when they, in development, that's how they migrate in, and well, they just got stuck there and that's where they stayed. That turns out not to be true. There are some there, but the great majority are farther down the heart, down the surface of the heart, but just located on one wall on the posterior surface. So that's a big surprise. And so that's the second surprise I'll save until I show you a little later. These are the bulk of the neurons. They're way down from the base of the heart. They're all distributed all the way down the full length of the left atrium, all the way to the juncture with the ventricle. So that was a big surprise. The other thing that we wanted to find out was how consistent is this? Is this distribution unique to this rat? It's not too helpful if it is, but as you're going to see, it turns out it's pretty damn consistent. So you actually have a structure, a consistent structure that you can attack to find out its function and connectivity and begin to try to influence specifically with pharmacological interventions, molecular interventions, electrical interventions, and possibly do something good. Here's just taking individual tissue sections of what the sections look like, where the neurons are within the specific tissue sections. And so we did some quantitative clustering to build the basis for being able to compare across hearts. And here's the distribution of the clusters, the things that are put together by their packing density and then positioned within the 3D space of the heart. And so here's the aorta, and here are the different clusters. And then here they are laid out on a 2D landscape. Then, oh, this is the female heart, sorry. It's slightly different. We'll talk about that in a minute. But then we did more clusters. Now we're getting to the molecular biology. So we sampled neurons, pulled them out, and did RNA-seq on them in order to get the picture of their full gene expression profiles. What are all the different genes that are expressed in each neuron and where are they located? And this analysis just shows that there are molecular phenotypes that have specific identities and that cluster together in a gradient within the structure of this so-called enterocardiac neuronal system. So that gives you molecular targets that are specific as well. So that's the story. And I'm going to show you, I hope, a movie. So you can appreciate the distribution of cells in this one male heart.
Is there any sound with the movie? Because we're not hearing anything.
No, there isn't any sound. There's no voiceover. You just have to make it up as you go along.
Thank you.
So now we did 3 more male hearts and 3 female hearts. And this is what that looks like, I hope. And so we did a really detailed spatial analysis, and the hearts strongly overlap. They're very similar distribution. The difference in the female heart likewise has a pretty similar distribution. The clusters are not quite as dense. There are not as many neurons per cluster. So there are fewer neurons overall, but the overall distribution is pretty similar, which kind of correlates with the physiology that female hearts have less strong baroreceptor vagal reflexes. The influence of the vagus on the female heart is a little less strong. That might be because there's fewer neurons. So I don't know if you, I hope you can appreciate that, but that's the story. That was the big interest. It's not
Are,
just.
are the male hearts to the left and the female to the right, or are the male hearts at the top and the female at the bottom, or?
The four males are at the top, and the female hearts are at the bottom, and their neurons are pink. And there's one more movie. If this one stops, you're supposed to be appreciating that the distributions are in the same part of the heart, even across male and female. So that was a pretty shocking finding. Most people really were surprised that the neurons extend and as extensive as they are, have as many clusters as they do, are as consistent as they are. These have been big surprises in the community. People had no idea. So our collaborator in Burlington, Vermont is behind the software for the graphics. And we made the figures, and we and people in Florida did the mapping. And there's more that I'm not going to be able to show you. There's a mathematical scaffold made by people in New Zealand. All this stuff's been mounted in the scaffold for quantitative overlays. So you can see exactly what the overlays are. So there's a lot going on with this.
Jim, can I ask a question?
Sure.
What are the implications of having more neurons than you thought? I mean, in terms of the physiology as opposed to, you know, or what are the implications, would you say?
They're distributed a lot more widely, and I think that provides you a lot of specific and selective opportunities for intervention. If they had been variable in their distribution and clustered all in more or less the same place wherever they lumped and happened to be, you wouldn't have really a way to approach them in any differential way. So here you got a chance to really pull out different clusters and and do something selectively and try to find the one you want. But what we did, and I'm not going to tell you about it, this is a threat, not a promise. I'm not going to tell you about it. But what we've done, John Gorky, who some of you may remember, my MD PhD student who gave the talk on what your poo is telling you and things like that, that kid. So what he did, and what's really cool, is he figured out how the heart is, what the heart is doing to these neurons to be protective. And I can go through a whole bunch of slides, but it's too much. It's too late, and it's going to kill you. But in very brief, it's a molecular biology approach. And so it rests on gene expression and looking at the interactions of the microRNA and the mRNA. And a lot of the, by looking at so microRNA regulate the activity of mRNA. So the mRNA, as you know, that's the transcribed part, and that's what's supposed to make the protein. But as it happens, it's heavily regulated by microRNA. And so why does the vagus grow inactive, and what causes it to be active? Well, here's the strangest story you ever want to hear. I promise I'm not going to show you these slides. Stop. But it turns out there's this thing called ischemic preconditioning. It is the bizarrest thing that somebody found out. Ischemia is where you eliminate the blood supply to some structure for a period of time. And So if you do, and John did is he gave ischemic conditioning to hind limbs by tying off the femoral arteries for a period. Then he released them and the flow was restored. And then he cruelly went in and gave the rat a heart attack, cut off its arterial supply to its left ventricle, gave it a nasty heart attack. And if there had been ischemic preconditioning, the heart was not damaged. It protected, the ischemic preconditioning protected the heart, absolutely. I could show you the slides, but I won't. And it depends on the dorsimotor nucleus and the vagus nerve. If you screw with them, you don't get it. You have to have an intact dorsimotor nucleus and vagus. That's where it comes from. So what is the influence of ischemic preconditioning on this vagal activity, both at the dorsimotor nucleus and down at the heart at the ICN? And that's what John's been working on. He's turned up all kinds of candidates. So our next thing is to try to intervene with microRNAs and see if we can, with those, reverse cardiac damage, in which case we're on the road.
Jim, can we go back a little bit about the difference between the male and the female vagus nerve? Would that account for some of the reasons why the heart attack? All the studies have shown what male, a lot of what we know is what the male reaction to a heart attack is, and females tend to have the silent heart attack. Well, would that help explain some of that?
It's a little more mysterious than that, because if females have lower incidence of heart disease and heart attack when they're premenopausal, when they have estrogen. But once they hit like 65, their incidence is pretty damn similar to males. And these are all young rats. I don't know what happens to their ICNs. They can change. But, you know, I think it's hormonal, maybe more than a neuronal. But nobody actually knows. It's obviously a topic of great interest. Females have not been studied much. It's mostly all male studies throughout the NIH. The NIH is trying to reverse that now. They require male and female subjects in all their studies now. So we're beginning to learn a great deal more about female anatomy and physiology and disease. But yeah, premenopausal female are very well protected. They rarely have heart disease or heart attack. So hang on to that. Keep good faith.
Thank you, but what about postmenopausal? That's the problem.
Fat and work out a lot. Don't get fat and work out a lot. Same as a guy.
Well, I do a lot of the workout, but
yeah. I think it'd be better. I'd be much happier for us guys to learn the secret of premenopausal women than the other way around.
Yeah, but once you hit menopause, you're still there.
I know, but how's the after? If it's hormonal, how is it protective? Exactly. So this Gorky guy again is deeply into all these different factors that interact with the myocardium and the neurons, and he's a total mysterian on the business of circulating inflammatory molecules. He really thinks that's the secret to everything. He's going to be a fantastic physician researcher. So it might be that the smaller number of neurons is useful because, you know, one thing, here's a hypothesis for you. Since the vagus nerve, so the vagus nerve has such a powerful effect on the heart, it's crazy. If you stimulate the vagus nerve, you can stop the heart cold. I mean, just stop it, not another beat. So that's pretty profound. And that's true of, and so the vagus nerve regulates the heart activity on a beat-to-beat basis. It's not fooling around. It's not waiting for an hour to see what's going on. Every heartbeat is carefully sculpted by vagal activity. And if it doesn't like what it's seeing, it'll stop it cold. Like if you get really scared and you freeze, your heart stops. And so, so the question is that true of females as well? I don't think anybody's ever looked. It's certainly true of males. So if females have a great deal of less, maybe as much as half less, maybe 30 or 50% less total neurons at the heart. Can they also have that profound a regulation? And it might be that heart disease is due to the fact that male hearts are so overstimulated and overregulated. You know, every little bump in the road sets your heart off in some crazy direction, and maybe it wears it out, beats it up. How's that for a hypothesis? Certainly, it's very feasible for this low vagal tone and the cardiac disease that comes with it to be a consequence of stress, where there's a big, profound, so I was telling you how these cranial nerve nuclei, both the sensory and motor sides, can be traceable, connected to other parts of the brain. And a massive connection is to so-called limbic structures that are involved with emotional state. In particular, amygdala, which has a big effect on stress and fear and things like that. And so if you're living with a lot of stress and fear and upsetness, and that drive is coming down onto the vagal outflow, that could have a big effect on your heart. One place where I'm pretty sure it does is in alcohol withdrawal. So if you're a true alcoholic and you suddenly decide to stop drinking, that's a bad idea. It's a bad plan. You know, don't stop. Taper off. Get some help. But if you go cold turkey, you really need to be hospitalized, because the emotional lability coming out of the amygdala drives crazy arrhythmias through the vagus nerve and kills you in a pretty good fraction of cases. If you're going to go cold turkey, you probably should be hospitalized. In fact, you should be hospitalized. To keep an eye on all that. The therapy to it is benzos. But we had a case not long ago that I read about. So if you're taking like alprazolam or Xanax or whatever, and you're taking like 0.25 milligrams or something like that, that's probably a reasonable dose. There's a guy in the ER that they gave him 30 milligrams and he still was throwing arrhythmias like crazy. Tough business. The opiate withdrawal is really unpleasant, but it doesn't tend to be fatal. But alcohol can be very damaging. And it's through that pathway, through the vagal production of arrhythmias.
I'm sorry. I have a question if you have time, Jim.
Sure.
So it's my understanding that the vagus nerve provides more information from the body to the brain than the brain to the body. I don't know that that's true, but it's what I've read. And there's something very important. If you could just talk a little bit about the stimulation of the vagus nerve, because there's a really strong movement in yoga and in sleep meditation to use a stimulation of the vagus nerve. And what ends up happening is that it's mostly done in stimulations through chants and singing and humming, which may have other connections to Arden, but if you could just speak to that type of connection and if you know anything about that work.
Yeah, I'll tell you a crazy story. So when I was a graduate student, I worked with rabbits because they were bigger and I wanted to stimulate the vagus nerve. And I built these cuffs to stimulate the vagus nerve in awake rabbits. So they were silicon plastics, they were real soft, and I could, put another soft metal lining on the inside of them to stimulate and run them out the rabbit's back of his head through under the skin. And we got so many crazy effects from that all over the brain. It was quite profound. And one day I was in what's now the Super Fresh in Society Hill, and a friend of mine from Penn, at that time I was on the faculty of Penn, and a friend of mine on the faculty of Penn, who was a neurologist, came up to me. His name was Sal Arakar, and he said, "Teach me how to stimulate the vagus nerve in a live patient." I thought he was nuts. And but he wasn't. He was serious. And he went on to build a whole company using these methods I developed as a graduate student.
They all are female patients.
And then he developed ways to stimulate the vagus externally with a, right, push it up against your vagus on your neck, blast it away. And his company was very successful. And now lots of people are doing that. My friend Michael Oshinsky has got asthma, and he's learned how to, give himself a few doses over time and stop the asthma. And you are quite right. The vagus is about 80% sensory or afferent, but most of it's the gut. There's a huge intercourse with the gut, pretty much with the heart, lungs, larynx, pharynx. And in the gut, it's more and more gets turned up all the time. It didn't used to be seen that liver was influenced, but now liver is actually pretty important, and certainly stomach and small intestine and gut microbiome and all these candidates. I don't mean to be obscene, but it looks like female orgasm depends on an intact vagus. So I don't know why that's the case, but there's literature now that claims that. So
you're saying if females have more sex, their vagus nerve is better?
Exactly. If you're enjoying it, you got a good vagus nerve. So, but I don't know why that's the case. Is that bizarre? But there's so many things that get published that I don't know what to believe anymore. The vagus has got so much attention as, influence in meditation, influence on pain, influence in epilepsy. Yeah, it's certainly a thing. And more broadly, I would just cast it in this little broader way that where we started out by talking about the brain is in your head, but it doesn't do anything unless it's talking to everything else. And the interaction with the organ systems is critical to mood, to back and forth. It's a two-way street. A visceral, emotional, neuraxis. It's not one only causing the other. They're going back and forth. I think if you had a bad ticker, you'd probably be in a pretty bad mood, and vice versa. If you're in a pretty bad mood, it might give you a bad ticker.
What impact of, like, for example, the right or the left bundle branch block? If you had a complete bundle branch block on the right or the left, how would that impact the vagus nerve?
It doesn't impact the vagus nerve. That's at the heart. But there is sidedness to it. The right and left vagus nerve and right and left innervation of the heart are different, and the diseases at the heart are different. So I have a lot of colleagues that are at UCLA. We're part of this Spark team, and part of the Spark team is at UCLA, and they're cardiologists, and we're learning from each other all the time about this, trying to figure this stuff out. It's mostly still blank slate. Nobody knew about this distribution of neurons. This was available to study and work with. So hopefully we'll get somewhere and answer some of these mysteries. But Carrie, I mean, you're reflecting a huge literature, as you know, that talks about this. The vagus nerve is not a homogeneous nerve. It's comprised of many little fascicles or bundles that are distinct, and many different fiber sizes. There's big, heavily myelinated A fibers that conduct really fast, that are mostly sensory fibers, and mostly from lung stretch and other things where it's important to get the information in a timely way. And some so-called B or B minus fibers that are lightly myelinated and a little slower. A lot of the innervation of the heart that controls the rhythm of the heart is B fibers. But the vast bulk of the vagus nerve are C fibers, which are slow, unmyelinated, get there when it get there kind of fibers.
But it's also running from the brain. The vagus nerve runs from the brain all the way to the tailbone, and and having that, I think it has this impact on all of the organs that we were talking about. Thinking about the parasympathetic nervous system and digestion and these connections to the vagus nerve, I think are really important to recognize, because I think that if you can learn to, again, and I know we're talking about generalizations in stimulation, but stimulating it in a way that the vagus nerve can help you to stop this fight, flight, panic thing that sometimes happens. I think that these are connections between the impact on the heart. And I know it impacts other parts of the body, but I think that this is really important research for lots of different reasons.
Yeah, we think so. I really have been a champion of that idea that there's this interplay, this visceral emotional neuraxis, and the role of the vagus nerve in coordinating peripheral activity. And so the same John Gorky I keep talking about, we just wrote a paper that makes the claim that the vagus nerve is not just a motor nerve, it's actually endocrine nerve. It's a coordinator of a parasympathetic endocrine system. So if you have a sympathetic endocrine system that involves the adrenals and hypothalamic pituitary axis and so on. But if you look at the gut, it makes a lot more peptides and modulatory factors of all different kinds that are distributed all over the place in the gut in huge amounts. I could not tell you the cells, but it doesn't matter. And the vagus gets information from them and turns it around and regulates them, and it coordinates those patterns. And that's effectively a parasympathetic endocrine system. And those circulating hormones, can profoundly affect health. And so being able to monitor them, intervene with them, regulate them, just like we do the adrenals and so on, is maybe in the future. As you can tell, I miss this guy.
So Jim, can you talk a bit more about how the heart's brain is independent, how it might have its own thoughts?
Like, yeah, there's two phenomena, and one has a Japanese name, and the other I can't remember the name of it, but I should. But they're probably both products of these neurons. And so one of them is if you have heartbreak, if you're disappointed in love or something, people can have profound effects, and they arise from this neural system. People can really go into a decline. And the other thing is, and I can't remember the name of it, is a wasting that occurs with heart disease, where some decent fraction of patients, maybe like 30%, if you get heart disease, no matter what you eat, you waste away. You become terribly thin. I'll show you a picture. You want to see something awful looking. Oh, come on, John. Where is it? I didn't know it was so deep in here. I'm sorry. It's quite a profound picture. These patients, here it is. You see that?
Yes.
So, it's all connected together into big systems. That looks pretty bad, huh? Carrie, you ever seem to know a lot about the vagus. What got you interested in all this?
Yeah, a personal story. And people who know me know this, I work in a really high stress job, and my company brought in a yoga instructor. It was phenomenal, changed my life, and now I have a home practice. But what happened was I'm having a lot of difficulty sleeping, and one of the solutions was to try to turn off your brain, and it doesn't work for me very well. So I looked to a deeper practice in yoga, and some people had recommended meditation. So during some of my meditation studies, I read about the vagus nerve. So I'm not a student of it at all. I'm not a scientist. I just believe that what's working for me, I would love to have other scientists or other people at least share ways to improve our lives overall. I don't, again, I'm not an expert in any way, shape, or form, but I think that it's also sometimes people think it's like kooky too, right? That you can dance and sing and enjoy music. People know my husband's in the Ardent Singers, and it's exciting to us to celebrate music and particularly at a time like this. We're in a very high, we've never had anything like this in our world where people are, mandatory masks and stayed away from each other. You can't hug your people, and you got to be social distance. So there's a lot of stress going on. So for me, it's been that type of a thing to look for other solutions to how to deal with your stress. So that's where my study came from. It wasn't because I got an A in biology. I did not.
Well, you seem to have found out a lot about it, and you should be a big fan of the Spark program because it's doing what you want it to do. It's trying hard, and there are different phases of it. This phase that I was initially part of was very foundational, but the next phase is going to be much more oriented towards disease and function. And so, it's going to keep on going and hopefully do some good. But about the COVID thing, my MD PhD students have been talking about the admissions to the rapid increase in substance abuse and deaths.
Yes.
Under COVID.
Yes, and absolutely.
Jim, I have a question. I'm curious to go back to the difference between the male and female hearts. You said the neurons were bundled differently and there were more of them.
No, the neurons themselves, they're similarly bundled. They have similar clusters, but the clusters are less densely packed. They're not as tight together and make quite as many neurons in the clusters.
Do you have any sense of how they function differently and why there is that sexual disparity?
Well, there is that finding that the stimulation of the vagus nerve does not have as profound effect on the heart in female as male.
Okay.
Same kind of effect, but not as strong.
Right.
You had mentioned, or there in the introduction, there had been a suggestion that there might be some things that you could do to improve the vagus input to improve the how the heart works and the control of the heart.
That's Spark. That's what we're trying to do. That's the goal.
Okay, so you don't have any suggestions tonight that you could just kind of like toss out to us?
The answer to everything that people ask like that I ever hear from anybody in practice is, or any study, what the highest thing you can do?
Wait about 10 years.
No, physical exercise is by far the best thing you can do for yourself.
Okay, so if you do cardiovascular exercise, then there's not a whole lot more you can do.
Well, there are, like, you can try to enhance immune function, but the immune function is also influenced by exercise. But the a lot of people I know who are coming in contact with patients are really making sure their vitamin D is solid, their vitamin C is solid, and that they're taking zinc, and they take zinc lozenges under their tongue to try to stabilize the epithelium of the nose, and
So I was recently diagnosed with a complete left bundle branch block, and
oh no,
I was and my doctor's like, oh, do you have a lot of pain in your chest? I'm like, don't have pain in my chest, and I exercise. I get, I have a exercise routine that I've been doing for years, where it's like 500 calories on an elliptical machine on Mondays, Tuesdays and Thursdays are bike rides, Wednesdays and Sundays are a core, abdominal exercise, and then Fridays are my swim, and Saturdays we walk or do something like that. And I'm like, and that, well, doesn't your chest hurt? It's like, I did I recently had an echocardiogram stress test, and the doctor kept asking me the whole time, are you okay? Do you have, is your chest hurting? Are you okay? I'm like, no, I'm fine,
That's fantastic.
Yeah, but they apparently my heart was still beating abnormality, more abnormal than what they would expect from a complete left bundle branch block. So
This is what they're seeing in the echocardiogram?
yes.
The nature of the contraction. Uh-huh.
Well, apparently the chamber, or the divider between the left and the right was just, it wasn't contracting when the left and the right ventricles, it was just flowing back and forth. So.
Wow. Did they decide, do they think there's some damage or some scarring or something? Or
I have another test coming up on the 27th where they're going to do a cardiac CAT scan. But I mean, it's like today I did a bike ride and I had my heart rate up to 171. So
nice. Wow, that's crazy.
I do a good cardiovascular exercise when I work out.
I'd say you do. Do they look with ultrasound at all? Do they do that? To see if
they had the ultrasound as part of the echocardiogram stress test. Yeah.
Yeah. Then they couldn't see anything structurally.
No.
That's pretty interesting. Well,
the kicker is my dad died of a heart attack at 58. So.
Uh-oh. And I hope you're premenopausal, right?
No, I'm past that.
Damn. You want a cardiac consult, or are you happy with you guys are seeing?
I could always welcome a second opinion.
Yeah.
Do you have somebody to suggest, or would you be?
No, I mean, I would talk to the people you have and say, where's the best cardiology group around here? Is it Penn, Jeff, Hopkins? If you're surrounded by quaternary care top tier groups, where would they like a second opinion? I would work with them. I wouldn't go around them.
Okay. Well, sometimes I always, I always feel like people, that they have a buddy system and they're not going to go against what their friends say. And if you go to somebody else that's not related, you get a true second opinion.
I don't think a good doctor should be like that. A good doctor should want you to have the best second opinion possible.
Okay. Thank you.
Okay, that was that we can transition to something else that is of concern, and that is Dominic is asking, Jim, have you found any link between the vagus nerve and vascular dementia, or can you speak to that at all?
That's out of left field. I don't even know what vascular dementia is. Dominic, what is it?
Okay, this is his wife. I'm sorry. It's a form of dementia caused by heart disease. My father, well, it was said that he had it. He had a bundle branch block before he was diagnosed with it. So that question popped into my head.
Wow. I wonder if that sounds like what we were talking about, in which cardiac dysfunction can affect mood and brain function.
Yeah, and he did have a lot of wasting away at the end of his life, but he had a lot of other problems too, so it's hard to say. But yeah, so.
And what was the form of the dementia? Did he have classical psychiatric symptoms, or just?
Yeah, pretty much. Yeah, pretty much. But it was kind of sudden. It was like he didn't have dementia that lasted over like many years. It only lasted, I mean, as far as we could tell, maybe 7 years. So that was part of the thing. Was it was such a sudden, more or less a sudden thing that came about. And then he had the heart condition as well.
So is it your sense that the brain issues were subsequent to the heart issues, or?
Yeah, I mean, of course, I'm not a doctor, but it seemed like there was definitely a connection, and then he was diagnosed with that. So anyway.
Yeah, I'm really, it's really intriguing what you say. I didn't know about that, but I'm going to learn more about it and see if I can see if there's a literature on it.
Okay. Okay. Well, thanks for your time. Thank you.
If I learn something, I'll point you towards it.
Jim, do you think you could change the slide, please, to something else?
I'm sorry.
That's okay. There, that's fine. Yeah. Thank you.
I'm sorry. Yeah, that's
No,
pretty.
it's okay.
When you work with sick stuff, you get insensitive to that. This is what John did with the remote ischemic preconditioning, where with a myocardial infarction, you get this tremendous damage in the top rows, and then the bottom rows you add ischemic preconditioning, and then add MI, and you didn't get such an effect. The hearts were pretty protected. Probably is also gruesome to you guys. Sorry. So, thank you for being somewhat interested in this. I found that your questions are interesting and good.
Yeah, thank you, Jim. This is great. Appreciate it.
Yeah, I guess I'm still having fun.
Would you stop the screen share, or I can do that if you like.
Hold on.
So.
If I can find the mouse, there it is. Hey.
There we are. I just
Hello.
wanted to say thank you to you and everybody so that we can all see each other for a minute. And don't forget to donate to the Arden Club if you can. And I just want to thank Jim for his time and expertise, and thanks for being our neighborhood scientist. So thank you.
Yeah, thank you. One thing I wanted to say is people are always asking me, have you retired, or are you thinking of retiring? And then I should say I am retired, right? I did retire.
This is your retirement.
This is my retirement. People get hobbies. Some people garden or woodwork or do pottery or poetry. I guess this is my retirement activity.
It sounds very intriguing.
Keeps me interested.
It's always best to do what you're most passionate about for as long as you can.
Yeah.
That's great.
Well, you know what I really like is it helps me a lot with this COVID stuff, which I find pretty dismaying, and the uncertainty around it, the lack of ability to really pin down facts and what to do and how to actually keep yourself. Everybody I know who's gotten sick they were being careful as hell. They just got sick. They have no idea how. It's scary. And some people get really sick. And so that's distraught. That's distressing. But every day we have a scrum meeting at 9 o'clock in the morning. With the team comes together and we all boost each other up and talk about how it's going and what we need help with and stuff like that. And that's very uplifting to me. Really, really helpful.
Is everybody still? Is everybody remote, or does everybody go into the office?
Oh no, the people go in, but you can only have one person to a room so far, and so there has to be some scheduling or rotating. I've never gone in since March, and they pretty much have told those of us who are You can do it remotely. You can do Zoom and write. We don't have to do it in the lab. Do it at home. They don't want us in there. So, you know, I've got my wife here who's doing telemedicine with her psychiatric patients. And then we get in different parts of the house and we're both Zooming away, blah, blah, blah. It's really weird. Very weird. We get together for lunch. That's nice. Never got to do that before.
All right. Well, that is terrific. And we're going to say goodnight. And don't forget that hopefully in a couple of days, I'll be able to have this recorded and put up on the, a link put up on the Arden Club website. So any other questions before we go? I'd like to just say thank you to everybody for coming, and we're excited that this worked out. So thank you, Jen.
When you donate, is there an automatic joining of the Arden Club, or is there a membership fee? Or
there is a membership you can join, and that's a separate fee. And I can't remember how much it is. It's not a lot. It's something like $25, $30 a year. And you get benefits like you get, you know, discounts off of tickets and all of that. But then you can just donate. Also, there's a separate link. So I think it's ardenclub.org/donate. So, all right. Thanks everybody for coming. Thank you so much, Jim.
Thank you, Jim. Thank you, everybody.
Hey, Nellie, if you stay back for just a minute, I want to tell you why I suggested that.
Okay. Thank you.
Thank you.
Bye.
Bye now.
No.
Do
a dinner sometime.
Someday.
The reason I brought it up is just because, you know, not every cardiologist is identical to every other cardiologist. People have different ideas and different approaches and different knowledge. And some people are expert in some things and some people are expert in other things. And sometimes if you have a weird thing going on, like what you have going on, you need to shop around some and see if you can make a connection somewhere.
Right. Well, so yeah, they wanted to put me on a beta blocker, and I'm like, you know, I know there's a correlation between beta blockers and dementia. There's also a correlation between beta blockers and an increased risk of death if you get the COVID-19. And there's other concerns. And I also tend to, whenever there's any adverse reactions with any drugs, I tend to get, you know, I get them. And then, so I don't take a lot of drugs because I don't react well with them.
So you know what blockers do, right? You have an idea about what they are?
I do regulatory writing, but it's yes and no.
So norepinephrine, epinephrine, you know, those are catecholaminergic transmitters that are ubiquitous in the brain and in the body. The sympathetic nervous system uses them, and many transmitters in the brain rest on them. And so there are different receptor types that catecholamines access, and beta receptors is one of them. Beta receptors happen to be heavily distributed on the heart. Alpha receptors are heavily distributed on the blood vessels, for example.
Mm-hmm.
So a guy named Ira Black in England discovered beta blockers as a way to control arrhythmias some time ago. And so it's been out there for a while. There's a huge background of literature on it. There's also a huge background of literature on side effects. And so it's not without risk. And
I think one of the main, you know, I had my echocardiogram stress test where my heart, my blood pressure was really, really high. It was like 140 something, 146 over like 92, 93, 94, 96, you know, throughout the test. But that was the day before my mother's funeral. You know, it was a really tough time. And then knowing that I've got this heart problem on top of the whole thing, I think, you know.
Yeah, well, it sounds like you have an unusual condition, and that's why I thought you might want to see if you could make a match to somebody who knew a lot about it.
Right.
as far as beta blockers go, you know, they're relatively innocuous for healthy people. It's famous that like violinists take them to keep their hands from trembling on stage. And
Well, you want a little bit of that tremble just to give that little that tone.
not so much. So it's something
I play viola, so
I would, you know, if you're not really suffering with anything that needs treatment, I wouldn't take drugs. But I'm just thinking, you know, it sounds like you have a weird thing going on, and
told you I'd have he'd jump in my lap at some point.
you might find somebody who's knows a lot about it that can help you, or at least help you know what to expect.
Thank you. Thank you.
Are you from the Forward School?
That's my husband's. Yeah.
It's my ancestors.
Ah, our kids went to Forward School. Thank you very much.
I didn't do too much. I was just born.
That's a good thing to do.
Yeah.
It beats the alternatives, right?
Yeah. All right.
The family line.
I just wanted to tell you, I wasn't trying to criticize your doctors or anything. I just was thinking that's a weird thing, and you know, might.
Well, I think, you know, that they want to do some more studies. They want to do some more testing. But I mean, he told me to take a baby aspirin every day, which I'm okay with doing that. So I'm not going to take the beta blocker unless I'm become completely convinced that I need to take it.
That sounds smart. I guess they're worried with arrhythmias. Like, I guess this is a kind of arrhythmia. There's a higher incidence of the possibility of throwing clots, which might be giving you little strokes.
Right.
And so I guess that's the baby aspirin idea.
But what the abnormality is that I think that they saw, and he was explaining it to the technician when I was sitting right there during the, you know, during the test, is that it seems like the wall between the left and the right ventricle, the septum, isn't contracting. So when the right ventricle contracts, the wall blows out that way, and then when the left contracts right after it, it blows out the other way.
Yeah, so it's supposed to contract itself and shorten, and it isn't.
Right, and
So-
and I think that's what they were concerned that's the abnormality that they're seeing in my heart, and they want to get to see if there's something that's causing that. So that's why they're doing.
Yep, that's good. All right, I'll let you go. Thank you.
All right, thank you very much. Thank you. Excellent talk. I really appreciate it.
Thank you so much.